Hi friends, hope you are fine. In this video, we have 30 questions on cell cycle, mitosis and meiosis. Without wasting a second, let's begin. Which of the following is the longest stage in the cell cycle? Options are A. Anaphase, B. Prophase, C. Metaphase, D. Interphase. You have 10 seconds to answer. The correct answer is interphase. Interphase is the longest phase, consists of G1S and G2 phase. And this is a scorecard. Please comment your score at the end. Question number 2. What is the primary purpose of mitosis? Options are to produce genetic diversity, to replicate DNA, to divide the nucleus and its contents, to facilitate cellular respiration. Correct answer is to divide the nucleus and its contents. In mitosis, the main purpose of mitosis is to divide the nucleus and its contents equally between two daughter cells, ensuring that each cell receives a complete set of chromosomes. Question number 3. During which phase of cell cycle does DNA replication occur? Options are A. G1 phase, B. S phase, C. G2 phase, D. M phase. Correct answer is S phase. DNA replication takes place during the S phase of the cell cycle. During this phase, the cell duplicates its genetic material in preparation for mitosis or cell division. Question number 4. What is the correct order of the phases in the cell cycle? Options are A. G1S G2M, B. M G1S G2, C. S G2M G1, D. G2MS G1. The correct answer is G1S G2M. And that is the order of the phases in the cell cycle. This sequence ensures that the cell grows, replicates its DNA in S phase, and prepares for division before undergoing mitosis in G2 phase. Question number 5. What is the role of cyclins in the cell cycle? Option A, to repair damaged DNA. Option B, to regulate the progression of the cell cycle. Option C, to provide energy for cell division. Option D, to facilitate protein synthesis. The correct answer is to regulate the progression of the cell cycle. Cyclins are proteins that bind to and activate cyclin-dependent kinases or CDKs, which are essential for the progression of the cell cycle. It helps regulate the timing and order of the various phases. Question number 6. Which of the following checkpoints occurs during the G1 phase? Options are A. DNA damage checkpoint. Option B. Spindle assembly checkpoint. Option C. DNA replication checkpoint. Option D. G2 checkpoint. The correct answer is DNA damage checkpoint. DNA damage checkpoint occurs during the G1 phase of cell cycle, ensuring that DNA damage is repaired before the cell proceeds to the S phase and replicates its DNA. Question number 7. How many chromosomes are present in a human somatic cell after mitosis? Options are A23, B46, C92, D24. Correct answer is B46. Human somatic cells are diploid. That means they have two sets of chromosomes. That is 46 chromosomes. After mitosis, each daughter cell will receive a complete set of 46 chromosomes. Question number 8. What is the main difference between mitosis and meiosis? Option A. Mitosis produces 2 genetically identical cells, while meiosis produces 4 genetically distinct cells. B. Mitosis occurs in gametes while meiosis occurs in somatic cells. C. Mitosis involves 2 rounds of division while meiosis involves 1. And D. Mitosis produces haploid cells while meiosis produces diploid cells. The correct answer is mitosis produces two genetically identical cells, while meiosis produces four genetically distinct cells. Question number 9. What is the purpose of metaphase plate during mitosis? Option A, to separate sister chromatids. Option B, to align chromosomes for separation. Option C, to replicate DNA. Option D, to form the nuclear envelope. The correct answer is to align chromosomes for separation. Metaphase plate, as you see here, it is an imaginary line at the center of the cell. This alignment ensures that chromosomes are properly positioned for separation during anaphase. Question number 10. During which phase of meiosis does crossing over occur? Options are A, prophase 1, B, metaphase 1, C, anaphase 1, D, telophase 2. The correct answer is prophase 1. Crossing over is the exchange of genetic material between non-sister chromatids of homologous chromosomes that occurs during prophase 1 of meiosis. This process increases genetic diversity. Question number 11. What is the outcome of meiosis? Option A, 2 diploid daughter cells. Option B, 4 haploid gametes. Option C, 1 diploid cell. Option D, 2 haploid cells. The answer is 4 haploid gametes. The outcome of meiosis is 4 genetically distinct haploid daughter cells or gametes, as you see here. Question number 12. Which of the following processes increases genetic diversity during meiosis? Option A, DNA replication. Option B, cytokinesis. Option C, crossing over. Option D, chromosome segregation. The answer is crossing over. Crossing over during prophase 1 of meiosis increases genetic diversity by exchange of segments between non-sister chromatids of homologous chromosomes. This creates new combinations of alleles in the resulting gametes, thus increases genetic diversity. Question number 13. What structure holds sister chromatids together? Option A, centromere. Option B, kinetochore. Option C, chromatin. Option D, spindle fibers. The answer is centromere. Centromere, as you see here, it is a structure that holds the sister chromatids together until they are separated during anaphase. It is a constricted region where the kinetochores assemble and where the spindle fibers attach to the chromosomes. Question number 14. Which phase of mitosis is characterized by the alignment of chromosomes along the equatorial plane? Options are A, prophase, B, metaphase, C, anaphase, D, telophase. The answer is metaphase. During metaphase of mitosis, the chromosomes align along the metaphase plate, which is an imaginary line at the equatorial plane of the cell, as you see here. Question number 15. What is the primary function of spindle apparatus? Option A, to replicate DNA. Option B, to separate chromosomes during cell division. Option C, to synthesize proteins. Option D, to form the nuclear envelope. The correct answer is to separate chromosomes during cell division. The spindle fibers attach to the kinetochores of the chromosomes and pull the sister chromatids apart during anaphase, ensuring that each daughter cell receives a complete set of chromosomes, as you see in this figure. Question number 16. What is the main difference between anaphase 1 of meiosis and anaphase of mitosis? Option A, anaphase 1 separates sister chromatids while anaphase separates homologous chromosomes. Option B, anaphase 1 separates homologous chromosomes while anaphase of mitosis separates sister chromatids. Option C, anaphase 1 occurs in somatic cells, while anaphase of mitosis occurs in gametes. Option D, there is no difference, they are the same process. The answer is anaphase 1 separates homologous chromosomes, while anaphase separates sister chromatids. As you see here, this is a separation of sister chromatids that is happening in mitosis, where homologous chromosomes are separated in the anaphase 1 of meiosis. Question number 17. What is the role of the G2 checkpoint? Options are A, to ensure DNA is replicated correctly, B, to check for spindle fiber formation, C, to assess cell size and nutrient availability, D, to monitor chromosome alignment. The correct answer is to assess cell size and nutrient availability. The G2 checkpoint ensures that the cell has grown to an appropriate size and has sufficient nutrients and growth factors to proceed with cell division, and also checks for any DNA damage. Question number 18. What's the term for the physical division of cytoplasm during cell division? Option A, mitosis. Option B, cytokinesis. Option C, interphase. Option D, telophase. The answer is cytokinesis. It is a physical division of the cytoplasm after mitosis or meiosis. It results in the formation of two or more daughter cells, each with its own nucleus and cytoplasm, as you see in this figure. Question number 19. Which of the following is a characteristic of cancer cells? Option A, they have a longer cell cycle. Option B, they undergo apoptosis more frequently. Option C, they can divide indefinitely. Option D, they do not respond to growth factors. The correct answer is they can divide indefinitely. One of the hallmarks of cancer cells is their ability to divide indefinitely without undergoing normal growth arrest or apoptosis. This uncontrolled cell division is a result of genetic mutations that disrupt the cell cycle regulatory mechanisms. Question number 20. What is the main function of telomeres? Option A, to protect the ends of chromosomes from degradation. Option B, to facilitate DNA replication. Option C, to hold sister chromatids together. Option D, to assist in protein synthesis. The correct answer is to protect the ends of chromosomes from degradation. Telomeres are repetitive DNA sequences located at the ends of chromosomes. The major function is to protect the chromosomes from degradation and fusion, ensuring the stability and integrity of the genetic material during cell division. Question number 21. In which phase of the cell cycle does the cell grow and carry out normal functions? Options are A, G1 phase, B, S phase, C, G2 phase, D, M phase. The correct answer is G1 phase. G1 phase or GAP1 phase is when cell grows and carries out its normal functions. It is the first phase of interphase during which the cell increases in size, synthesizes proteins and organelles in preparation for DNA replication in the synthetic phase or S phase. Question number 22. What is the significance of the synaptonemal complex during meiosis? Options are A, it helps in the separation of sister chromatids. B, it facilitates the pairing of homologous chromosomes. C, it assists in DNA replication. D, it forms the spindle apparatus. The correct answer is it facilitates the pairing of homologous chromosomes. It's a protein structure that forms between homologous chromosomes during prophase 1 of meiosis. It helps align the homologs and facilitates the process of crossing over, which increases genetic diversity in the resulting gametes. Question number 23. How many times does a cell divide during meiosis? Options are A, once, B, twice, C 3 times, D 4 times. The correct answer is twice. Meiosis involves two rounds of cell division, meiosis 1 and meiosis 2. During meiosis 1, homologous chromosomes are separated, while during meiosis 2, sister chromatids are separated. This results in the production of 4 genetically distinct haploid daughter cells from a single diploid parent cell. Question number 24. What is the main purpose of the anaphase promoting complex, or APC? Options are A, to initiate DNA replication, B, to trigger the separation of sister chromatids, C, to repair damaged DNA, D, to align chromosomes at the metaphase plate. The correct answer is to trigger the separation of sister chromatids. The anaphase promoting complex is a protein complex that triggers the separation of sister chromatids during anaphase of mitosis and meiosis. It does this by making certain proteins for degradation, allowing the cell to progress from metaphase to anaphase. Question number 25. What happens during telophase? Options are A, chromosomes condense, B, nuclear envelope reforms, C, chromosomes align at the metaphase plate, D, sister chromatids are pulled apart. The correct answer is the nuclear envelope reforms. During telophase, the nuclear envelope reforms around the separated chromosomes at each pole of the cell. This marks the end of mitosis or meiosis 2 and the beginning of cytokinesis, the division of cytoplasm. Question number 26. What is the primary function of the kinetochore? Options are A, attach spindle fibers to chromosomes, B, to hold sister chromatids together, C, to facilitate DNA replication, D, to form the nuclear envelope. The correct answer is to attach spindle fibers to chromosomes. Its primary function is to serve as an attachment site for the spindle fibers during cell division, ensuring the proper segregation of chromosomes to the daughter cells. Question number 27. Which of the following statements is true about homologous chromosomes? Options are A, they are identical copies of each other, B, they carry the same genes but may have different alleles, C, they are produced during mitosis, D, they are always found in haploid cells. The correct answer is they carry the same genes but may have different alleles. Homologous chromosomes are pairs of chromosomes that carries the same genes but may have different alleles, as you see here. They are found in diploid cells and are separated during meiosis 1 to produce haploid gametes. Question number 28. What is the role of checkpoints in the cell cycle? Option A, to ensure that the cell is ready to proceed to the next phase. Option B, to promote rapid cell division. Option C, to repair damaged DNA. Option D, to facilitate protein synthesis. The answer is to ensure that the cell is ready to proceed to the next phase. Cell cycle checkpoints check for proper DNA replication, chromosome segregation, and cell size before allowing the cell to progress to the next phase. Question number 29. During which phase of meiosis do homologous chromosomes separate? Option A, prophase 1. Option B, metaphase 1. Option C, anaphase 1. Option D, telophase 2. The correct answer is anaphase 1. This reduction division ensures that each daughter cell receives one chromosome from each homologous pair, resulting in haploid cells. And the final question, what is the consequence of errors during meiosis? Option A, increased genetic stability. Option B, genetic disorders or aneuploidy. Option C, enhanced DNA repair. Option D, reduced cell division rates. The correct answer is genetic disorders or aneuploidy. Errors during meiosis, such as non-distinction or improper chromosome segregation, that lead to genetic disorders or aneuploidy, which is the presence of an abnormal number of chromosomes in a cell. This can result in conditions like Down syndrome, Turner syndromes, etc. Comment your score below. Take care, stay blessed. Thank you so much. You are with biologyexams4u.com.